Bismillahirrahmanirrahim. Assalamu alaikum. This is Amna Mehmood and I'm going to tell you about the types of RNA. There are two types of RNA that are genetic RNA and non-genetic RNA. Genetic RNA. Genetic RNA is found only in some viruses where genetic material DNA is absent. It means that RNA will be the genetic material for them, such as viruses. It means it is not present in eukaryotic cells. The RNA present in all eukaryotic cells are non-genetic RNA. Thus, we conclude that the genetic RNA is only found in viruses and not found in human beings or the eukaryotic cells. Coming towards the non-genetic RNA, it includes messenger RNA, ribosomal RNA, and transfer RNA. Messenger RNA, the other name of messenger RNA is hnRNA. HN means heterogeneous nuclear RNA, which means that it is found in the nucleus. Ribosomal RNA, there is no other name for the ribosomal RNA, whereas the other name of transfer RNA is snRNA. SN means small nuclear RNA. The other name of transfer RNA is also called soluble RNA. Messenger RNA is single stranded with no folds and no loops. It is a straight chain, whereas ribosomal RNA is also single stranded but it has folds or loops. Transfer RNA is also single stranded with arms and loops. The molecular weight of messenger RNA is 5 lakh daltons, whereas the molecular weight of ribosomal RNA is 40,000 to 1 lakh daltons, whereas the transfer RNA has molecular weight of 23,000 to 30,000 daltons. The messenger RNA constitute 3 to 5% of the total RNA, whereas the ribosomal RNA constitute 80% of the total RNA and transfer RNA possesses 10 to 20% of total RNA, which means that the amount of ribosomal RNA is maximum, whereas the amount of messenger RNA is minimum. From the molecular weight, we can conclude that the messenger RNA is the largest kind of RNA, whereas the transfer RNA is the smallest type of RNA. Coming towards the production, the messenger RNA is synthesized from the DNA. The ribosomal RNA is involved in protein synthesis and the transfer RNA bring the amino acids at the site of protein synthesis. The transfer RNA, by the name it is understood that it transfers something. It transfers the active amino acids from one place to the site of protein synthesis, that is the ribosomes. Next, here in the diagram you can see that these are codons AUG. AUG is always the starting codon. It is known as the starting codon, and whereas UAA and all that, these are called stop codons. These are also known as nonsense codons. Whereas if we discuss about the ribosomal RNA, it provides the binding site and decode the messenger RNA. Here is the structure of the ribosomal RNA. As you can see that there are also folds and loops present here and it is not a straight chain. Whereas messenger RNA was a straight chain. It provides binding sites. For example, if the messenger RNA that is produced by the DNA have a nitrogenous base known as adenine, so adenine will form bond with thiamine, or in case of RNA, adenine will form bond with uracil. Got it? Then uracil will, if uracil is present, then adenine will be its comparative, and if the cytosine is present, then guanine will be its comparative. So it decodes messenger RNA and provide the binding site. Transfer RNA, hairpin model and clover leaf model. It will be a very lengthy topic if I will discuss this. Then we come towards the overview of protein synthesis. As you can see that first is messenger RNA. Messenger RNA is produced by the DNA. It contains all the information that is necessary to produce the protein. The information is given to it by the DNA and the information is known as codon or cryptogram. Protein synthesis needs specific information that is delivered to the messenger RNA by the DNA. Next, coming towards the ribosomal RNA, here you can see that it is of spiral shape. There are also loops and there are folds and there are hydrogen bonds between them. One is adenine, so the comparative is uracil, and one is guanine, so the comparative is cytosine. So here are the bonds. There will be two bonds between adenine and uracil and three bonds between cytosine and guanine. It will decode the information on messenger RNA and transfer RNA, transfer the amino acids and to the ribosomes, or you can say that the site of uh, reaction or transcription, and it will be form protein. Thank you so much.